Hello and welcome to the Osteoarthritis Action Alliance Lunch and Learn webinar for November 19th, 2025. Thank you for joining us today, and thank you to the current Osteoarthritis Action Alliance sponsors who support our work, which includes the monthly Lunch and Learn series. Now it is my pleasure to introduce today's speaker, Katherine P. Liao. Dr. Liao is a rheumatologist and clinical investigator in the Division of Rheumatology, Inflammation, and Immunity at Brigham and Women's Hospital, and associate professor of medicine and biomedical informatics at Harvard Medical School. At Brigham and Women's, she directs the NIH-funded Core Center for Clinical Research known as the VERITY Bioinformatics Core. Dr. Liao holds a joint appointment at the Veterans Affairs Boston Healthcare System, where she co-directs the Applied Bioinformatics Core serving the VA Million Veteran Program and the Massachusetts Veterans Epidemiology Research and Information Center. Her research includes patient-oriented research studies, as well as integrating state-of-the-art bioinformatics approaches, incorporating electronic health record data to conduct studies aimed at improving our understanding and management of human diseases, with a clinical focus on rheumatoid arthritis. The title of today's presentation is Bioinformatics for Getting EHR Data AI Ready for Clinical Rheumatology Research. Welcome, Dr. Liao.
Great. Thank you, Katie, and thanks so much for the kind introduction. I realized as you were reading the title that I probably should have had ChatGPT help make me a more snazzy title, but we will move forward. So I'd like to start with an image. And I believe this image might, for many of you, immediately evoke, you'll know that this is a bald eagle. And perhaps you might not even remember when you learned a bald eagle. It's just something that you know, and that when you see it, you know it. Here's another bird. This is our state bird in Massachusetts. It's a black-capped chickadee. Some of you may not have seen it, but I imagine that if I were to show it to you again, you'd immediately know it's a bird and try to remember that it's a black-capped chickadee and that it's different from a bald eagle. And so this is hoping to give you a sense of what's the difference between natural and artificial intelligence. So natural intelligence is something created by nature, natural evolutionary mechanisms. Biologic intelligence, what's hardwired in the brain, or any including, this is in one of the definitions out there, hypothetical alien intelligence. But contrast that to artificial intelligence, which is technology that enables computers and machines to simulate human intelligence and problem-solving capabilities. So it's not in and of itself, doesn't have common sense, but we can train it or tell it, give it general guidelines on what it is we're trying to do. So for a human, for us, looking at these images, we know immediately it's a bird, and we can tell they're two different birds. But for AI, it would mean taking apart these images into pixels, processing them, understanding the patterns in the data, and understanding the differences between these two patterns, which can be done relatively quickly, but it's not the same kind of intelligence that we have, which is kind of this built-in natural intelligence. And I'll give some more examples of that later. So as we move into this field of using more and more AI, particularly in my focus today will be on clinical research and electronic health record data, there is this interface that I am starting to, we don't think as much about, and it's that interface between human and AI. How do we interact with AI? How do we use it more as a tool? How do we use it as a tool effectively for what we're trying to do? So I'm a clinical researcher, and this is a highly reductionist view of clinical research. So what we try to do is understand the biologic or clinical significance of factors that relates to outcomes, particularly health outcomes that we care about. A very simplistic view here is looking at the association between for example, an independent variable, such as a treatment intervention, and a dependent variable, such as an outcome, such as osteoarthritis or total knee replacement. And I'd like to just set this framework there because we will be using this framework over and over as we go through the talk. The other piece of why we've been talking so much about AI, particularly in clinical research, is this recent explosion, literally, of clinical data that's available. So I'd say concurrent with with AI, or actually together, we've also had a lot of big data that has become available that wasn't available before. And part of that has to do with our shift from paper charts to electronic health records. So when I was in training, we were still using paper charts. So when a patient came in with a heart attack and he says, Doc, I was here 3 years ago. I had a heart attack and I had a stent, but I'm not sure I remember what happened. So we would have to order the charts, go to the basement, flip through a stack of charts over the past 3 years and figure out exactly what happened. So you can imagine the amount of time it takes for that one patient. That would be very difficult to scale up to large populations. And so it wasn't common to have large population-based studies. There were large population-based studies, but very few of them. Fast forward to today. So in 2009, the US government enacted the HITECH Act, which incentivized institutions to adopt electronic health records for care. And it worked very well. There was an exponential adoption of EHRs. And now we are drowning in the data. As a clinician, I know I can find it in there somewhere, but now the issue is the data are there. I can't find where it is and where the relevant data are. And in fact, EHRs are transactional systems. They aren't even designed for patient care, and they're obviously suboptimal for research. However, they are capturing every transaction that happens between a patient and the healthcare system. And it does enable now these studies in large populations, because not only is it capturing whether a patient was seen, but it's capturing also the test results and all the things that the clinicians are writing about what they think the patient has. So I'd like to step back, though, and talk about some of the standard, like very tried and true types of study designs, of which prospective observational cohort studies are one of them. And this is, when I mentioned before with the chart review, it's very hard to scale up. There were very established, very powerful observational cohort studies that were available, such as Nurses Health, such as the Framingham Heart Study. And I can't not mention the Johnson County Osteoarthritis Project, that really helped us understand a lot about risk factors and outcomes of disease. And so prospective, many of you probably already know, obviously know what a prospective observational cohort study is. You are following a population of patients forward, but the power of these design is that you are assessing for all the patients the same clinical assessments or other assessment at regular time intervals. So, for example, in this figure here, you can see at the baseline time interval, you're assessing risk factors, both the presence and the absence for all the patients or all the participants, and also assessing all the outcomes for all the participants so that you don't have this ascertainment bias. Now, where EHRs and other kinds of real-world data are helpful is that sometimes even the best designed observational cohort study doesn't have all the risk factors that maybe something new came up. You discovered there's a new disease. There are things that you can't possibly think of when you started the study. And the power of EHRs is that you are capturing all types of factors, clinical factors and outcomes and phenotypes as part of routine care. However, what you don't have is this very standardized assessment of risk factors on all the patients at all times. So you don't know if, let's say, there's no mention of osteoarthritis, you kind of need to assume that patient didn't have osteoarthritis, but you don't actually know that they don't have osteoarthritis. So there are limitations. So I'm offering this up as an alternative study design or something to complement all the milieu of wonderful clinical study designs we have out there. So I mentioned we're drowning the data. The data are there. They're potentially powerful, but how do we effectively use these data? So today I'd like to focus on maybe the simplest approach, which is designing a cohort. If you want that cohort to study, so I mentioned these very well-established observational cohort studies, can we possibly have something similar to that with this real-world electronic health record data? And step one is to define the patients that you're interested in. Who is the cohort? And so what I thought I'd do in this slide is at a high level, help you think about if you're interested in using EHR for a cohort study, what do you need to think about before you even start it? And I'd say the first thing is, what is the main one or two clinical questions you want to ask or answer? And understand, and then take a look at the scope and size of the population you have. Are you looking at hundreds to thousands, tens to millions of patients? And are the key variables, or what we call many use in machine learning features, needed to address the question available? Are they codified? Are they ICD codes in there? Are they in the notes? If the data are not there, then just stop right there. You've got to go look somewhere else. Step two is defining the study population. So now you have your question. What is a feasible population for you to study? Is your condition very rare? Is it a small population? Should you just, is it something that the only way you can get it is chart review? Think about, are the codified data good enough? Some conditions, the ICD codes are quite good. They have a high positive predictive value. And you can use, for example, two ICD codes in a medication and have a good capture of all the patients. In most cases, and I can say now, having done this for quite many years, always checking to see if the ICDs can get by. We often need in our rheumatic and musculoskeletal conditions a combination of ICD and additional relevant data from the notes, for which we use natural language processing and machine learning. And I will talk talk briefly about both of those methods. The next concept I'd like to talk about is the phenotype, and I think we all use this word all the time, so I'd like to actually define it in more detail because it becomes very important as we think about defining and creating a cohort based on a phenotype. So phenotype is the foundation of clinical care and research. On the clinical side, we have to truly understand what it is we're treating. What is the phenotype, which then helps us construct what is the management of this condition. What type of, what's the flavor of RA or OA that we think we're looking at? What's the best management? On the research side, you want to very much create this parallel because in research, we're not seeing the patient in the clinic and we don't have all the same kinds of data, but we have similar data, particularly with electronic health record data. We will have similar data available to us to then construct that phenotype that we care about to define the cohort and the outcomes of interest. So what is a phenotype? And I thought this was actually best said by Dr. David Felsen, who I know is a giant in the field of osteoarthritis. And so in his paper here, he states phenotype is a characteristic observable by the naked eye, images, or blood tests. So something that's measurable that can differentiate one individual from another. And in contrast, a genotype is often the source of phenotypic variation, but it's only relevant to phenotypes when it creates an observable difference between two organisms with the same disease or two individuals with the same disease. Let's go back to talk about electronic health record data. And there are two main types of EHR data. And actually, these are two main types of data in general. There are structured data and there are unstructured data. Structured data are data that are hard-coded, that are standardized in their coding. For example, ICD codes, CPT codes, electronic prescriptions. Unstructured data is what they sound like. It could be anywhere and everything. Most commonly text data. It's what we're writing in our notes, what we're dictating. So there's not a clear structure. Not everyone is saying things the same way. Ultimately, at the end of the day, for most of our research projects, we need an analytic data set. We need the data in a structured format, which you can see on the right. That is mainly, it could be numbers, it could be, female or male, but it has to be something that is hard-coded and that you can analyze in an algorithm or equation. And so the structured data are well adjusted. They're well formatted for that. In the most case, they're messy, but you could write a code and extract the structured data. And you can see that there in the figure is the unstructured data that are all over the place. And so we have now available to us tools such as, and actually a whole field of natural language processing that allows us to parse the data, parse the notes into, and extract the concepts that we're interested in. For example, for rheumatoid arthritis, we're very interested in smoking and bone erosions. And so you can parse these notes to identify whether there's a mention of smoking or a mention of erosion in the notes and abstract that into a data in a structured format that we can use for research. So I'll just emphasize that table on the right is a theme to what the types of data that we often need to feed into AI algorithms or AI tools. So let's take go a little bit higher up and take the 1,000-foot view. What are we trying to accomplish here? Well, we've got our EHR data. We've got lots of data in there. We would like to do a project on, as I mentioned, that very simplistic design of perhaps risk factors and outcome for osteoarthritis. To do that, first, you need to identify who has osteoarthritis. And starting with the EHR, which often contains millions of patients, you can extract the structured data and unstructured data and process the unstructured data into a format that is then structured that you can analyze together. The second step is very important. It's defining your phenotype. You want to know that the phenotype that you're classifying is a phenotype that's important for clinical care. And then you can move through the steps of algorithm development, which I will go through later, but very broadly, you develop the algorithm and you test it on the labeled set, or you train it with the label set to make sure the algorithm is classifying patients you believe have osteoarthritis. You evaluate the algorithm, and when you apply the algorithm to the cohort, you can then get that cohort of patients with osteoarthritis to then do your downstream application studies. So this is, I do think that we have come a long way in our ability to really leverage these large amounts of data to answer questions we might not have been able to answer before because the data were there, but they were embedded in the notes or in a format that we couldn't study. However, there are also limitations to some of these approaches. So what I'd like to do is take a moment and show you a real life blooper on a phenotyping project we had on myocardial infarction. So myocardial infarction is an outcome that many of us study. And so we created an algorithm. We developed a pipeline for the algorithm in Boston, Massachusetts. And we ported this algorithm to Nashville, Tennessee, an institution in Nashville, Tennessee, to Vanderbilt, and as well as Northwestern in Chicago, Illinois. The algorithm performed fine. Then we took this algorithm or this pipeline to the VA, the Veterans Affairs, their national healthcare system, lots of data in there. On the top panel, we created an algorithm with ICD codes and NLP, so data, MI codes extracted using NLP. On the bottom is the map of MI prevalence if we only use ICD codes. And I'll give you a moment to take a guess at what is wrong. And unfortunately, I can't see the chat, but I'm happy if people want to put it in because I'm curious to see if anybody sees this. So for those of you who are good on your US geography, you'll notice that on the upper panel, you'll see that there's something going on with Michigan. The prevalence of MI is much higher in Michigan than the rest of the country. And as some of you may have guessed, the reason for that is because the abbreviation for Michigan is also MI. So while we tested the algorithm in multiple institutions and places around the country before taking it to nationwide VA, we did not have Michigan in there. And so this just shows you some of the many different things. One is that you really need to test and always validate your algorithms in different places, but also that natural intelligence allows you to immediately see that there's something going on there and kind of know what's happening, like this is MI, as opposed to artificial intelligence, which can run very complex algorithms, but will not necessarily be able to identify, have common sense to say, oh, something's going on here. Okay, so with that in mind, I'd like to go through some of the ingredients for a phenotype algorithm, and I will go through a brief example, or maybe not specifically about OA, but take you through some of the steps that we might think about for osteoarthritis. And I'm using an example of an actual project that's happening right now. I've been very fortunate to have the opportunity to work with Amanda Nelson, who's at the University of North Carolina, also the PI of the Johnston County OA project, and Jeff Curtis, who's at the University of Alabama at Birmingham. And so together, our groups are working toward creating an OA cohort. For us, it's for us at the Brigham, my group, the Bioinformatics Core, is a new challenge to assist in at a large scale, develop a cohort outside of our system using the pipelines that we have when our team is at the VA or at the Brigham. But what are the ingredients for a phenotype algorithm? Number one, you need domain expert knowledge. You need somebody who really understands that phenotype and creates not just those gold standard labels, but looks at all the inputs into the algorithm. So for example, what are we going to say is important features or variables for the algorithm? Look at the output. The data must have some informative data. So I mentioned the EHR data are vast, but you can't have vast data with no data, no information. What we think less about is the importance of data management and processing of these data into a format that's clean and QC'd for all the downstream applications, whether it's the algorithm or actually the association studies. In the middle, I've glommed everything together. If you remember that blue box I had right in the middle, epidemiology, math statistics, computer science, among others, to really summarize the data, analyze it, validate it, and interpret the results. And finally, the last piece that we often don't think about and often take for granted is with the increase in capability of storage and processing power that we now have. So in the next few slides, I'm going to just take you briefly through some of the methods we have available or some of the tools we have available and concepts behind what's available when we're thinking about phenotyping. The first step, I think, that most people are stuck at is what EHR features or variables should we even consider for an OA algorithm? as clinicians, we know what goes in, but we really are thinking just about codes and medications. But with AI, there's an opportunity to think about a lot more of those informative features to have a bigger, have a more robust algorithm that can really capture your patients at a higher sensitivity and specificity. So what I'm going to show you here is this is a knowledge graph of all the codes that are associated with osteoarthritis in the EHR of the VA and Mass General Brigham. And what we have here is a resource now as a starting point for people who are maybe trying to understand, well, what should I care about with osteoarthritis? Or what are things that I might not have think about that may be important for osteoarthritis? Or in this case, what are the features we should input into an algorithm that we're thinking about for OA? And so and I mention this also because it's the AI can do a lot of things, but you need to get the data in very discrete formats. It can take a lot of data. And what this is now we're not just expanding the breadth of what we can do, but the amount of data we can consider when we're building an algorithm. Um, and so how do we get here? In the interest of time, I'm going to maybe go over this a little quicker than I was before. But back in 2013, there was this idea that this group at Google published this idea of word to vec. It's like taking relationships and changing them into vectors. So if you look on the left-hand side, man is to woman as king is to queen. They're not exactly the same, but their relationships are the same. But how do you connect them? How do you connect these two concepts to say that they're related? And so by changing it into vectors where you have both a distance and an angle, you're now effectively transforming that into a structure. It can be numbers. And once you have those numbers, you're able to take these concepts and compare them to each other at scale. So imagine doing this. So imagine taking all the concepts. So that's, for the EHR, that's ICD, electronic prescriptions and lab codes, and creating a matrix of how they're related to each other, creating vectors. And then as numbers, you can then look at how they're all related to each other. And that's how we come up with something like this, which is a knowledge graph. And this is actually the foundation for a lot of the large language models we have today. I'm going to fast forward here to how you might push this into a phenotyping system. I talked about the EHR. Now we've extracted a lot of the data into the EHR. Once you extract those data, you can create a covariance matrix that is then we have pipelines to take that large covariance matrix and use that to identify patients with and without OA. And I won't, with the interest of time, I'm not going to go into more detail here. Just that the ability to have access to the data, to process these data, to create these large detailed matrices has actually improved our ability to phenotype and understand to identify who has what condition or outcome in large data sets with more accuracy than you could with, for example, just ICD-9 codes. And so now I've taken you through the whole pipeline from the EHR all the way to the cohort and to the downstream application to where now, if you have an OA-based cohort, that's our EHR-based platform with the risk factors and outcomes laid on top of a computing infrastructure and informatics, which then allows you to do a very, using the same epidemiologic and biostatistic methods to look at the association between an independent variable and an outcome, such as total knee replacement, with a lot of still complex methods and study design considerations in between. So in summary, I hope that I've brought home to you this idea that it really is it's a perfect storm in a good way. The increased availability of clinical big data, advancements in methods and technology, informatics and computing that has allowed us to increase what's possible for clinical research. And the final common pathway for most of AI methods is that you do need the structured data, I put in quotes numbers, and that we've heavily relied on bioinformatics to get our data in an AI-ready format. And last but not least is the importance of maintaining the principles developed from epidemiology, biostats, and our prospective cohort studies to use in our observational studies with EHR data and AI. So AI is another module that we're inserting in, but integrating into the framework we already have. And last but not least, this is a photo of our combined VERITY Bioinformatics team. And what I'd like to hammer home is also that clinical research with big data and I is a complete team sport. And I'd like to thank my longtime collaborator in the middle, Tianchi Kai, who is professor of bioinformatics and biostatistics at Harvard Medical School and School of Public Health, the rest of the team, and our funding sources. So with that, I'd like to thank you for your time, and I'm happy to take any questions.
Thank you so much, Dr. Liao. That was fascinating. And I know you were just scratching the surface and you really boiled down a ton of information, just kind of like AI, into 25 minutes. So amazing. We do have time for a question, for a few questions. So if anyone in the audience has a question for Dr. Liao, feel free to type it into the chat. I'll just get us started here. Dr. Liao, you mentioned large language models, but that's probably what I'm most familiar with in terms of AI. And so I'm curious how to get the data into a format that, as you say, is AI ready. Can large language models help with that? And do they themselves need data in a certain format to then be processed?
Yeah, yeah. So large language models, as you all know, are very good with text data and large amounts of text data. And they're able to well summarize the text data. I do think that large language models will be very helpful in helping us get the data ready for AI, but you almost need an intermediate. They can summarize the data, but they don't know how to get the data into a format. So many of you probably heard about agents. And so they need some guidance to get them into perhaps, we're testing this down, I'm sure there are some examples that I missed out there, but they can potentially do that and help us there, particularly with the text data.
Great, thank you. And here's one that just came in. I'm assuming we can extrapolate digital phenotyping to other conditions and other models of data collection, such as wearable device data. Would we still use the same general concept?
Yeah, that's a good question. I think you absolutely can. And I think you have to use the same metrics that you use to evaluate whether the data are the right input. So, for example, wearable data, wearable device data. How good is the capture? What's the distribution? You just have to ask all the same questions. What are you trying to measure? And is that information in there? And what's because the wearable device will only be available in some individuals, you want to think about how you're using those data.
Thank you. It seems like, I mean, it just seems like the possibilities with this are limitless. So I'm curious kind of where you see your own research heading and what types of questions you'll be working on in the near future.
Yeah. What I showed you was the first step, creating a cohort, almost trying to, we can never have exactly the same as a prospective cohort studies with carefully collected clinical data, but can we create a platform for research? And so one of the things that we are doing is, as you all know, with clinical trials, they are very good at answering a very specific question. But there are some questions that will simply never be answered by clinical trials. And can we, with these tools, set up a platform to emulate clinical trials with real world data, with advanced phenotyping? Because one of the roadblocks to doing that is we don't have good phenotypes. We're not very precise in terms of who has what condition and getting to these subtype outcomes. For example, would be OA of the hip versus OA of the knee. So we are getting to that point where we can get more details, and I think that we can start answering those type of questions with real world data in a very structured platform with the right methods.
Great. And we're going to squeeze in one more question here because I think this is a really important one. How developed is the science behind the limitations and considerations researchers have to address when analyzing EHR data? I've observed a rush to analyze big EHR data, but without some investigators asking about how valid the analysis and what are the biases.
I think that is a very good question, and I think the science is not as developed with prospective cohort studies because EHRs really have not been available widely until recently. And I completely agree with the person asking the question. I think there needs to always be a pause, and just like any study, I think you just have to really look at the methods and have your eyes wide open when reading about the results.
Well, thank you so much, Dr. Liao. We will end it there. Thank you for a fantastic presentation, and thank you to everyone in attendance today. I'm going to launch a quick poll here. If you have time to answer a few questions before you leave, we'd appreciate it. And hope you'll join us on December 10th for our next Lunch and Learn, which will be titled Improving Muscle Health in Patients with Osteoarthritis and Obesity, Results from the Pomelo Trial, featuring Dr. Christine Gojiuk. Thank you all so much, and we hope you have a great rest of your day.
Thank you.